So yes, I'm going to speak about acute recordings with neuropixel probes. In the lab, we have 6 neuropixel probes, and we aim to record simultaneously from 6 visual areas: AM, PM, V1, LM, AR, and RL, but also subcortical visual areas such as LGN and LP. So here is our workflow. So we start by a pretty heavy surgery, which is a head post implant, a craniotomy, and a cover slip insertion. And we do that 4 weeks in advance compared to the recording day. And this is great because we avoid tissue damage, inflammation, and loss of blood on the day of the recording. Then the mouse goes through a health check for 10 days. And then if everything is okay, it goes to ISI and we map the visual areas. We check for anomalies in the maps. And then finally, the mouse can go for habituation with the behavior team, but also with the neuropixel team on our rigs. The mouse is then handoff ready. So we have a small surgery before the recordings. It's a removal of the cover slip, an implant of an insertion window, and a stabilization of the brain. And after 1 or 2 hours of recovery, we can insert the probes and record from the 6 visual areas. The mouse is then perfused and we clear the brain with iDisco method. We image it with optical projection tomography, and then we get the probe localization in CCF. So concerning craniotomy, so there are different approaches. So some just need one small craniotomy, like here. Some others would need multiple small craniotomies if the area they are interested in are separated. But in our case, we decided to do a 5 millimeter craniotomy because first of all, if we do a small craniotomy over each visual areas, it would overlap anyway. And also we wanted to have a good visualization of the vasculature. So this is the first surgery that I mentioned. So it's a head post implant and a cover slip implant. So we start by anesthetize the mouse, then we remove the hair, the skin, and we clean the skull. We put in place the head post with the well here, and then we metabond it to the skull. We start hatching the craniotomy and then removing the cap. We insert the glass cover slip and then seal it with quick cast all around. So here I have a video of the craniotomy, the 5 millimeter craniotomy and the durotomy. So I'm going to play this. And I wanted to say that for the surgeon, what is the most difficult apparently is that the thickness of the skull is not homogeneous around the visual areas. So it's pretty thick and posterior and pretty thin anterior. So here there is a damage, a potential damage with the drill. And then when the cap is loose, they will lift it from 3 o'clock and fold it at 9 o'clock. So you have a possible lift damage at 9 o'clock. So what they do is that they keep a hinge on at 9 o'clock. So you will see now they're lifting. And you can see that at 9 o'clock, there's still a little bit of something, and then it helps to not lacerate the brain. So you can even put ACSF on top, which makes the bone a little bit more softer, and then you would not lacerate the brain when you remove the cap. And if you have a big bleeding, the surgeon recommend that you use gel foam on the brain and a sugi to put a bit of pressure on it with cold ACSF. And now this durotomy, you see, it's pretty a work of art. She's using actually a method that we call the pizza method. So you go like different slices. And she's very skilled. Okay, so now we jump to the surgery that we do the day of the experiment. So we received the mouse with a glass cover slip that you can see in A. We have decided to use a suction cup to remove the glass cover slip. It costs maybe $1, and it eliminates every kind of differences between surgeon, and it eliminates also the use of forceps. So it's really practical. And here is a final result of that step in E. Then in F, we implant the insertion window. So it's a plastic transparent window with 6 holes, one for each probe, that we insert in the well here. We secure it with Metabond. And then the most important step here is to inject agarose between the brain and the window. That helps tremendously for the recordings, to stabilize the recordings. And then on top, we put a bit of silicone oil to avoid the agarose to dry. And then in J, here is the final product of the surgery. So here I have a video of the removal of the cover slip. So you see your cover slip here. It's attached to the skull with vet bond. It's a veterinary glue. And so let's play the video. So she will approach with the suction cup, make contact, put the vacuum on, put some ACSF, and then she will crack the window with a bent needle and lift it. So the the issues that can happen with this surgery is bleeding. So basically, you can have bleeding if you don't have a cover slip that is suitable for that. So we tested 4 different types of cover slip here. Some are glass only, PDMS coated glass, plastic only, or PDMS only. And we ranked the bleeding from severe to known. And you can see here it's very drastic that the PDMS coated glass is the one that triggers the less bleeding. So we stuck with this one. Also, with a bleeding, all you can do is having gel foam, and cold ACSF works really well. And then sometimes also you get bleeding because you have too much time between the 2 surgeries. So try to reduce that time. Now I have a video of the injection of agarose. So she comes with a blunt needles and go from the back and the front. And she decided that maybe there's not enough in the back, so puts a little bit more and clean it gently so that we have a smooth surface. The potential issues there are the agarose consistency, or having bubbles in the agarose. So what we recommend there is to just redo it, and with gentle steering, and also not in one shot the heating, multiple rounds of heating, and avoid boiling. When the agarose cools down too quickly, we decided to use to heat the syringe and the needle in the bath where the agarose stays, and it works very well. Also, after this step, we put some silicone oil. So the big problem is not enough silicone oil or too much. So you have to recover really all the agarose with it. And if you have too much, you remove it with a Sugi swab. Now we label the probes this way. So we have a head post here, and then an Eppendorf here that it's cut, and we glue it together. And basically we clamp this exactly where the mouse head would be, where the brain would be. And then we inject it with dye. Oh, by the way, we use CM dye because it's the only one that resists to the clearing, the iDisco clearing method that we have found at least. And then so we put the dye in there, and then we descend the probe one by one and remove them one by one. And here is a view of a top view of the probe in the dye on the rig. And obviously what we want is to see these beautiful tracks like it's in this picture. About grounding, so you saw that we have a well and we have a head post. So in the well, there is a cylinder here where you can insert a pin that is soldered to a silver wire. So here is the schematic. You can enter with the pin here, and the silver wire comes inside the well. So that's what you see in this picture here. The wall is in black all around, and you have the wire that comes on the side like this. And you can see also that after the surgery, this wire is under the window and in the agarros. One issue here could be that sometimes the wire can be broken, and in fact we turn to use like thicker ones, but we have a trick to add one from outside also and metabond it to the gold pin. Then you can clamp your mouse in the rig. So we use this tail guard here that protects the probe from the tail of the mouse. And then basically in this cone here, we have a golden pin here that mates with this golden pin here. And then we test with a multimeter. And then basically the probe ground becomes electrically conductive with the brain surface. So this is the insertion of the probe. So we go in 3 steps. First, we automatically go to the above the window. Then we go to the surface of the brain with small increment of 50 or 100 microns. And then finally, we go to the final destination at 200 micron a minute. And then what we like doing also is to do a retraction of 100 or 200 micron to release some tension. The issue with the insertion are, of course, you all know about that, membrane. So either the dura has not been removed or there's regrowth. So there are 3 things you can try. Either move the probes because it's maybe sometimes not uniform, so you can find a patch where it would work. You can also be just aggressive with the probes. They're bent enough. And then also the most efficient would be to use a sharpened probe. And of course, the only way to avoid bleeding on the surface is to avoid the vasculature and move the probes. And then if we have a problem with agarros drying and we see some probe drift, actually there's not enough silicone or there's bad application of agarros, and there's nothing to do in this case. So on the right here, we have a little graph about sharpened against unsharpened probes and the surface bleeding that we observe. So again, we ranked it from severe to none. Here on the right here is the summary for all the areas. And you can see that we don't see much difference in surface bleeding with these 2 types of probes. But of course, the sharpened probes are very useful to go through the dura. Here is an example of recording. We have receptor field, example receptor field for each area, rasters for many cells in the cortex, hippocampus, and thalamus. And we have a receptor field for LP and LGN in this recording as well. Just I want to mention that we generally record from more than 1,000 cells per session. And I wanted just to show you this quickly because I really like this video. So it's you can see the 6 different tracks of the probes in the cleared brain here. And that's it. Thank you.
Hi. Can you see my screen? Okay. So thank you, Severine. I'm going now to talk about a little bit different approach in performing acute recordings. In my approach, I do little craniotomies in multiple places. And before I will start, I just wanted to point when you use actually acute recordings technique. So you use it in head-fixed animals if you want to record large number of neurons from multiple brain regions. And this technique, it's not good for free moving animals experiments and recordings if you want to record the same neuron over days. And the main advantage of acute recordings is that you can record from multiple brain regions or multiple probes in the same even region. You have a lot of flexibility with insertion in angles. So you can plan your trajectory in the way that you will hit some brain structures that you are the most interested. And because Neuropixels probes, it's long, so you have it's very powerful here. And you can combine also with other techniques, for example, optogenetics or wide field imaging. The major disadvantage of this technique is that you can record only for a few days. You cannot track the same neuron over days, and the craniotomy quality decays over time, especially in the cortex. So now I'm going to walk you through the steps that I do for my recordings. I'm not going to talk about details about the surgery that was already discussed by Severin, and also there's many protocols available online that you can use to look at the detailed surgery. But what I wanted to highlight is that at the stage of head plate implantation of your animal, you should to think already where you are going to record from, because the design and location of the head plate implant will determine to which brain region you will have access to. And for example, here I'm showing pictures of head plate implantation using the International Brain Laboratory design. And if you implant posterior, you will gain access to very anterior parts of the brain and dorsal cortex, but you don't have so much access to cerebellum, whereas implanting this head plate anterior will allows you access to cerebellum and medulla, brain regions, and partly to dorsal cortex. What also I do in my at this stage, I also mark a future craniotomies. So the animal, it's on stereotax rig, so you have very high precision to mark where exactly your craniotomies should be, and you can use a surgical pen for that. So this is example of some of the future craniotomies that I was planning to do in this animal. And lastly, at this step, you don't do craniotomy yet, but you need to protect the skull from infection. So you can do either using, for example, optical adhesive glue, like in my case, and you need to just remember to cure this glue with the UV light. Or if you have other, you can also use other materials like Zapagap glue or adhesive resin cement. So the next step is to mount your probe so you can hold on your manipulator. So first you need to solder the connect to connect the ground and reference of the probe. I'm not going to talk about details here because there's a nice video on how to handle and solder probe prepared by Bill Karsh, so you can just watch how to do that. The next step is actually the mount the probe on the rod, and then depends what kind of rods you have. If you purchase from IMEC directly, your probes already have a dovetail cap that is attached to the probe, so you don't need to align. You don't need to glue and align this, and you can easily slide your probe on a rod that is also available when you purchase in IMEC. And you just need to tighten this with this little screw. If you choose other ways, for example, from New Scale Tech, or you purchase rods from Sensapex, you need to do this alignment by yourself. And especially in Sensapex, you need to additionally glue manually dovetail to the probe. And this is how it looks like a fully mounted probe with solder grounding wire. And my little tip is here that once you have mounted probe with the head stage, just remember to secure with a tape, otherwise the head stage will fall on the shank and it will be broken, and we want to avoid this. And the last step is to sharpen the probe to make insertion easier. And this is just a stone on which you are grinding the probe. and you can see that the tip of the probe really becomes sharp, and hence it's much easier to insert the probe through the dura, for example. The next, once this is done, you are almost ready to go, but still you need to check your setup. And the first thing that you should to do is to test your probes that you just mounted, if they are working. Test the noise level by measuring in saline, and if there are grounding issue, you need to debug these issues at this stage. So don't try to ignore the issue, because the issue won't disappear, so you need to debug. Then at this stage also you plan your precise probe trajectories. You've seen just several presentations how to do that, and you can already position your manipulators in the position that will allows you to reach the brain. And here I'm just showing a picture of my setup on which the manipulator is mounted on very thick rod from Thorlabs, but I use a magnetic base that allows me to easy and quick change of the position of the manipulator if this is needed. Also, what I use is this inclinometer to set the angle of the probe. So just remember to make sure that you can hit the target with the animal and the probe in place. If you cannot reach it, try to adjust the manipulator position or height, or you will need to change angle of the craniotomy, angle of the probe, and location of the craniotomy if this is needed. And once this is done, we are almost ready to do our first Neuropixels recordings. So we need to give access of the probes. We need to provide access for probes to be inserted inside the brain. So here I typically do craniotomies for my preparations around 3 hours before recordings, but you can do also a night before, and that depends on, of course, ethical protocol that it's in your lab. I do small craniotomies, like less 1 millimeter for single shank recordings, like for example this one, and some larger craniotomies if I plan a multiple probe recordings from the same craniotomy or from multi-shank probes. And the grounding pin, it's implanted and touches the brain surface, and usually it's in the cerebellum. But in some preparations, I need to implant somewhere else, otherwise, because I don't have so much space and access to cerebellum. So in this case, this grounding pin is implanted somewhere in somatosensory cortex. So once this is done, you can cover your craniotomies with silicone sealant, quick cast, and just don't remove this this silicone until you will really start your recordings. So with that, you have already animal prepared, so you are ready to go. So at the day of your recordings, first just prepare setup and manipulators for recordings, label the probes, and then connect the probes and see if they are detected by the software that you use. I use Spike GLX software that has been developed by Bill Karsh in Tim Harry's lab. And once this is everything it's working, you can head fix your animal on the rig and lower the probes, and then connect grounding pin to the probes. And as I mentioned in my preparation, the grounding pin, the golden grounding pin touches the brain, and I connect through these wires with the probe grounding pin that it's here. Then what I typically do, I go with the probe above the bregma, and I zero my manipulator positions above bregma. So then I can go really precisely to the location that I planned for my recordings. But if you are very confident with the place that you are recording, obviously you don't need to do this step. This is this is useful for multiple, for example, recordings from the same craniotomy, and it might be just easier later for you to distinguish which trajectories on histology belongs to which recordings. Once this is done, you go with the manipulator with the probe above your craniotomy, you zero the position of the probe that it's related to the depth at the surface of the brain. Just don't hit blood vessels. You want to avoid any bleeding. So whenever your probe or tip is hitting the blood vessels, just do a small adjustment so you don't hit blood vessels. And then start inserting the probes, typically with the speed around 5 to 10 micrometers. And always check if you can see already spikes in your software, in my case, Spike GLX. This is an example of raw data in Spike GLX from 2 probes that have been inserted simultaneously. And you should see very clearly the spiking activity once the probe is inside the brain and you think you reach the brain region especially if you reaches the brain region that's supposed to have a lot of spiking activity. So once the probes are inserted, you just wait 10 minutes and then you start your recordings. And afterwards, remember to clean your probes. And one small tip for recordings in a very deep structures, in areas that are close to the skull, like for example, hypothalamus or amygdala, or like lateral brain regions, for example, auditory cortex. So these are like some steps that I do when I have to hit this more difficult brain regions. So in this example, I was trying to record from auditory cortex, that it's very lateral, it's laterally located. So you insert the probe, and then around 800 micrometer before you should the tip of the probe hits the skull, you decrease, you start watching the probe. Then around 500 micrometer, you can decrease the speed of the manipulator to 3 micrometers per second, and then just watch when the probe starts bending. If probe starts bending like here, just stop manipulator, retract few micrometer until the probe looks again straight. And of course, just check your signal in Spike GLX that you didn't damage the probe or you didn't lose the signal in Spike GLX. And lastly, I will just briefly talk about most common issues during recordings. The first one is, of course, grinding. This is example of recordings that has huge noise, and because the grinding pin wasn't well connected. And this is what you should to see in your recordings. So nice spiking activity, not this noise. So if you have this, just first check connection with the probe wire, check other grounding wires. If it's needed, replace grounding wire because it can be broken. Try to connect grounding wires to the table or use different reference, for example, internal reference. Always identify sources of noise and try to ground this to the table. And also make sure that there's no leakage of saline or ACSF. The second common issue is that signal is saturated, or there's no signal, or there's some very weak spiking activity. So especially if you have no signal, restart Spike GLX. Sometimes there's some connection issues. Check cables, and as I mentioned, connections, especially with the head stage. Sometimes if you insert probe too fast, you might not have so many spikes. And this is example of the study that compared the speed of inserting the probe. And they show that if you insert probe too fast, you won't see like a lot of activity. Also may be related to damaged craniotomy or brain, or the probe might be bent or broken, or probe might be not cleaned properly if you see very weak spiking activity. And lastly, these are just my some suggestions how to maintain the craniotomy in this procedure. You can protect craniotomy with a thin layer of Duragel. In my recording, in my approach, I don't remove Dura because it's extra protection for the brain. I sharpen the probe that improves a lot the insertion process. Always record while craniotomy is fresh, and don't let the brain dry. And this is just the last slide with links of resources about acute recordings, some previous presentations, and this is just for you to check these links to get like more detailed information. And thank you for your attention.